Hi, class. Welcome to Experimental Psychology with Polly. Today, we're going to talk about the chapter 2 of Field and Holes, How to Design and Report Experiments. Chapter 2 is entitled Planning an Experiment. So let's begin. Here's the overview of the things that we're going to talk about. What is your research question? How do you get from general idea or interest into a specific testable research hypothesis? Then we move toward identifying, manipulating, and measuring your variables, and some considerations when you plan your experiment. So let's begin. What is your research question? So in this particular example, narrowing down your question into a testable hypothesis is a very challenging task to do. So if it is your first time to conceptualize a, a research, especially an experiment, it must be difficult to start from scratch. But you can start from somewhere. You can start from anywhere. For example, you can have your own area of interest that you would like to ponder on, that you would like to work on in your study. And with regards to the references where you can check and refer to some ideas, we can start from something general. For example, you identify your specific interest in this uh, process diagram. Let's say, Field, the author of one of the authors of the book, is interested in phobias. So he started with that idea. So if he refers to a regular textbook, most of the phobias that are featured on textbooks are animals. The subject matter of the phobias are animals. And then if he's going to refer to a specialist book, there will be more explanation as to how phobias among animals work. The mechanisms as to how it works might be presented there. So specialist book is actually difficult to look for if you're not familiar in the field that that you would like to work on. So a specialist book is like a compilation of different authors who are asked to write by the editor. So the experts who are the contributor of each chapter are considered to be like a summary of the different knowledge that has been known in a subject area. So a specialist book is not typically named like a usual textbook that we have, because in a textbook we have a very general and broad stroke on a branch of let's say science or a particular topic at hand. But for a specialist book, we are looking at a holistic, interdependent perspectives among the chapters, wherein it will provide uh, a story in different perspectives on that particular subject matter. Or it could be that phobia could be just one of the components that are included in a bigger picture in that specialist book. So let's say that specialist book might not be about phobia in general, but might be about anxiety disorders, and phobia is one of it. So it works like that. So in this particular example, he identified a specific specialist book. So you can choose maybe one or two chapters that you might uh, work on. You don't have to read the entire book. All you need to do is to focus on what you need. So focus on a particular area that you would like to study. So since the author narrowed down from phobias to animal phobias, he can work his way down to the details uh, about animal phobias, which is in this particular area, he's looking at the information process or learning of information about animals, and the perspective as to how do we take in this information. And then if this specialist book probably cited one of a journal review article or journal review articles, and then you will find several studies that provided evidence to that uh, particular subject matter. So in that case, it's you can consider that to be a backward uh, way of uh, reviewing the references, because you look at the cited materials that are mentioned in the particular material that you've read, and you want to read more of it because it was particularly important to the area of query that you're working on. And then from that journal review article, you can either read the cited materials there, or you can refer to Google Scholar and look at articles that cited that journal review article. So in that case, the the temporal reference is forward, wherein more advanced or more recent articles are written that cited that particular material that you're reading. So in any case, moving to the next level deeper, making it narrower, is another decision making path that you have to work on as you go along, as you read along, because identifying gaps in the literature is something that you can't really do in a superficial manner. You really need to think deep as you read the article, and you can generate your own questions as you go along, because the questions that you have in your head as you read will be the guiding would be the guideline as to how are you going to search for the next article that you're going to read. And you have to be very selective to the articles that you want to read if it captures the question that you are leading on. That's why if you have no improvement in the way you formulate your questions, then you can go all along without really accomplishing anything at all. You can read so much many articles without formulating a problem. So that's another skill that you really need to develop by polishing the questions as you go along and being selective to the articles that you're going to read. So you can determine if the article is within the scope of what you want to know. You can determine that through the title and the abstract. So when you read the abstract and you notice that you can make some connections in your head when you read this, then fine. But in terms of time management, you really need to work on try to curate which among the several articles that you've downloaded or flagged to be somewhat significant to what you need to do. You can determine the key papers, maybe about 3 to 5 papers, that will formulate what you need to work on. Or by having these key papers, they will be substantial enough in order for you to forward your argument or to forward the question that you have into a particular research hypothesis, testable hypothesis. Okay. And justifying a study or justifying an experiment is uh, another subject matter to work on, because you need to also uh, provide the rationale as to why an experiment is essential to move the discussion forward to the subject matter that you're working on. So in this case, uh, he was able to narrow down the query. If you notice the word counts, it becomes more and more detailed. So it means that it's becoming narrowed down into a particular perspective. And he funneled it down to this question, the research question: does fear information change children's fear beliefs? Okay. So what is your research question? So I ask you to listen to different podcasts, to whatever you find interesting, and perhaps you can also start from somewhere. You can start from your observation about the things around you, and the patterns that you notice that can make sense in trying to explain a particular outcome. So in this scientific statements and questions, you also need to determine here what are your independent and dependent variables, because in your research statement you have to be quick in terms of defining what your outcome is and defining what your antecedent event is. So I will leave you here to check and answer. You can pause from here, and you can check the answer on the next page. Okay, so let's move forward to the next part of our lecture: identifying, manipulating, and measuring your variables. Let's move along. Okay, what is my dependent variable? So it is a very simple yet fundamental and crucial part of the process when you plan your experiment. Because if you don't know your variables, it will be easy to get swayed, confused, and get get things disorganized along the way when you cannot really pinpoint what are your variables. So if you started with an unclear definition, identification and definition of your variables, then you are setting yourself to fail. So having this in mind is a crucial step before you can think of any other steps along the way. So in identifying your dependent variable, you must be able to justify the need for studying this particular outcome that you are worried about, that you are pondering on. Because if we are to expend our effort thinking about this particular outcome, then it must be useful for the others who will consume the research that you will come up with. So it should make sense that working on something should be useful to others as well. That's why justifying a particular subject matter should make sense not only for you but also for other people. Next, as we identify this particular outcome, of course, we try to think of antecedents or key factors that affect how this particular outcome variable changes. So in our example, we have here dependent variable as the children's fear beliefs, okay? And the independent variable here is the information change. So how do you then manipulate this particular independent variable? Okay, so it's difficult to think about how you're going to manipulate your IV if in the first place it's not clear to you what it is, how you define it, and what kind of conditions can you imagine in doing this. So remember in Mill's logic of scientific discovery, in manipulating the independent variable, we need to observe when it is present and when it is absent. So we can think of ways on how a particular independent variable is changed. And so if we observe the changes or the different forms or conditions of your IV, then we expect that the outcome will follow differently at a particular direction as well. Okay. So this is a very tricky part, and there's no one clear way on how to do it, because it also depends on the literature as to what are the crucial things that you should consider when you manipulate your independent variable. So there's no one size fits all in doing this. Okay. So it really uh, takes a lot of labor reading a lot of articles as to what aspects in your experimental design should be considered relative to what the literature says. Okay. So whatever experiment we're trying to do, there will be several ways in which we can do the same thing. So we can have the same independent variable, but we can think of multiple ways on how we can manipulate it. We can use several different, unique stimuli and different designs in our experiment, and we can still use the same independent variable of interest. So how tricky is that, isn't it? That's why you would require a lot of justification just to design an experiment. You need to consider a lot of things as you go along. So you need to be careful on the choice that you make, because every choice that you make can affect the result of your experiment. So I'm giving you a particular example of the exercise, or the replicated experiments of my past students. This is from Elliott, 2008, wherein they changed the background of the portrait, a female's portrait. So in this first experiment, I monitored this every step of the way, and this particular group did not report at all that I was not able to supervise them very closely because they were not reporting to me. So anyway, the error in their design is that the the grayscale description in the experiment refers to the photo and not with the background being gray. So that's the error in their design. And the reason as to why the photo is in grayscale is that perhaps the experimenters, the researchers, are trying to control for a variable about a person's attractiveness with regards to their skin color. So let's say having a more reddish skin would be perhaps more attractive. But in this case, all they're interested in looking for is whether the background red can actually enhance the judgment of attractiveness on the woman, wo- photo of the woman. So in this particular example, the manipulation here is the background. So again, presence of red and absence of red. Okay. So what happened here actually is that in this first set of the experiment, they find null results. And for this second set of the experiment, they find statistically significant effect wherein the red background is perceived to be more attractive. So the problem here, I suspect, of course, excluding that part, the issue that we already discussed in here, is that the stimuli, the stimulus used in the experiment should be pre-tested. So by pre-testing the stimuli, we can ensure that the stimuli is good enough to be able to come up with a variation of responses. So in this particular case, this photo showed to have some kind of a ceiling effect wherein almost everyone agrees that the lady is attractive regardless of the background color, right? So I'm thinking that perhaps the selection of the stimuli is another factor that we need to consider. And pre-testing of the stimuli also helps. So in this case, they did not do this exercise. And I suggest that in every experiment, uh, whenever we use stimuli, we need to pre-test it. And I must say that in the previous experiment that I conducted, I noticed that kind of bias wherein the selection of the stimuli was not particularly planned and pre-tested, and the results might have some kind of a caution when we try to interpret the findings, because the selection of the stimuli alone should already be systematic in the first place. Okay, so moving forward in manipulating the independent variable, so the author's topic has something to do with how fear information changes children's fear beliefs. So again, the independent variable here is the fear information. So what the author can do is to manipulate this fear information. So he thought that what kind of animal can we work on for children? And at first he thought about how about mythical creatures? So that could be the case. However, it's not ecologically valid. It does not reflect the real world. And then by addressing that issue, he thought of perhaps animals that are not familiar to most people, especially to children. So he identified three uh, animals in Australia. They are called coal, couscous, and coca. And these stimuli, they are chosen because it is assumed that children have no prior knowledge to this species. Okay, so perhaps we can also filter out any participant who are familiar to the to this stimuli, because we can easily do that. All we need to do is just to add that question: how familiar are they to the animal that was presented to them? Because having prior knowledge about them would already confound or tamper the information processing that is very crucial in the experiment. So they need to manipulate what kind of information can we deliver to children to determine to deliver it in a way that they can deliver negative or positive judgment. Okay, so the way they presented the info, so this is the manipulation of the independent variable. Positive, pertaining to them being cuddly and friendly and approachable. Then for negative info, defining them as animals who are dangerous and vicious. Okay. And another is no info at all. So that's the control variable. Okay. And then, of course, in that experiment, after presenting uh, these info, either whether they are presented with positive, negative, or control, then the children will have to respond to their fear beliefs about them. I will show you later on the questions that the author devised to measure that. So in this case, in manipulating the independent variable, you notice here that the animals were presented in different means of fear information. One is positive, one is negative, and the other is control. Okay, so you notice here that he jumbles around the order of this subject area, these conditions. We call them conditions. Okay, so we have positive, negative, and no information, and we have this trial items. We have coal, couscous, and coca. We jumble them all together in order to control for potential error in this kind of experiment. We can talk about that later on when we discuss the within subjects design experiment. So in the what I want you to focus right now is that the author used this technique called counterbalancing, okay, or some kind of a randomization in a way. So the purpose here is that if there is a systematic error derived from the order of these different trials, we are able to control them by scattering them equally along along the case, along each case. Okay, so there will be participants who will be presented in this of the experimental conditions in this manner, or in this manner, or in this manner. Okay, so moving forward, so how do you measure then your dependent variable? So in different experimental studies, there are many ways on how you can capture the dependent variable. So in most cases, for cognitive psych experiments, we have reaction times, or for neuropsych experiments, you can also capture physiological response. So what are those? It could be skin conductance, eye gaze, pupil dilation, heart rate. So all those things, those are physiological responses. Okay. Then self-report. So again, this is the very common means of capturing our dependent variable. Self-report, for example, we can use Likert scale or visual analog scale. Okay. Or maybe a dichotomous yes or no response can also work. So another is through behavioral measures, we provide a task that whatever your performance in that task might be representing a particular behavior, or or a decision making pattern. So your response to the task will be informative to the researcher's question. So it should be measuring a particular behavior. Okay, so finally, the last part of our discussion are some considerations when you design your experiment. So there are four major things in his book. So first is, how will I analyze my data? So there are different means to analyze your data. So we have parametric and non-parametric. And if we're going to refer to psychological research, most of them use parametric statistical analysis because relatively it is more powerful in terms of the classification of inferential tests compared to the non-parametric. So again, I want you to master this sequence, okay? So you really have to gain mastery on this sequence of thinking, okay? It's a process of thinking that can help you when you design your experiment. So first, you determine your hypothesis and lay out your independent variable. So you have to identify them and have a clear definition of it. Okay. So third is that determine the levels of the IV. It is related, of course, to number two by determining how many conditions you're going to have. So what are these conditions? And then determine the instruments used to measure, for example, your dependent variable. So when you also need to know how are you going to interpret this instrument. So let's say if you find a higher score on your dependent variable, what does that mean? If you get a lower score in your in your instrument measuring your dependent variable, what does that mean? How are you going to interpret that? So another is determining how the IVs are manipulated. Of course, that's already implied when we think about number two and three. Okay. And then number six. So again, for number five, that's very crucial, right? You need to be very careful by considering your research question in mind, your research hypothesis in mind. So because as you manipulate your IV, the design of the study is the next thing that you should think about. Okay. So yeah, so determining the experimental design of each IV. So because if you have more than one IV, you can have mixed design. Okay, one could be between subjects and the other could be within subjects. So it depends. Sometimes it also depends on the nature of the variable to determine what kind of design is more fitting. Okay, there are many things that you need to consider in the design. Then finally choose the appropriate statistical analysis to test the hypothesis of interest or the part of the analysis that answers your research question. Okay, let's move forward. Another thing that you need to consider is the validity of your measure in your of your variable. So in particular, we are referring to the psychometric properties of the instrument that you're going to use. So for content validity, we're testing whether the items in this scale are representative of the construct that you're trying to capture. For the criterion validity, we are interested to understand whether a particular measure predicts another outcome or another measure. For example, we have the admission exam score and then the GPA. So admission exam score is predictive of GPA. So it provides the admission committee some idea whether you can thrive or survive in the campus. So another is factorial validity. Again, we're interested in the psychometric properties of our scale. Let's say if you use a self-report scale measuring your dependent variable, we're interested to check whether the components that were mentioned in the literature are actually existing or observed, uh, whether it actually emerged in the data that you have now. Okay, because it's crucial when you interpret the outcome of your study if the psychometric properties are not as stable as you thought it should be. Okay. You need to also consider the reliability of your measure. So it has to have a consistent uh it has to have consistency because it will be difficult to interpret as well your analysis if you find out that the measure that you're using is not consistent. The typical measure that we use for the scale reliability is Cronbach's alpha. It's now easy to do it nowadays thanks to our software, statistical software wherein you just need some few clicks and there you go, voila. Okay. Anyway, you need to be careful with that as well. For example, if you have reversed items, you need to make sure that you were able to identify them before you conduct your reliability analysis, skill reliability analysis. Another thing that you should consider is the measurement error. So measurement error is the difference between the true measurement and the measurement that is captured by your instrument. So it's a perspective wherein we always assume that whatever instrument that we use, it will always have some kind of error. And our role as researchers is to minimize those measurement error. That's why we're interested in the psychometric properties of our scale. That's why we're interested in the reliability and validity of our scale. And we're also considering different means on how we can measure our dependent variable. Probably not only uh, considering self-reports, self-report formats, perhaps. We can also consider other means of capturing our dependent variable. So, but then again, it depends on the nature of the study, the nature of the variable, whether a particular measurement or instrument will be more appropriate than the other, or perhaps they could be measured altogether and see if they will converge as to one latent factor, referring just to one latent factor. We call that multivariate analysis. So that's for another subject area, okay? But uh, in particular, we can focus on just one continuous dependent variable or outcome. So here are some of the example of the dependent variable measures. So you can just look into it, okay? So we do not only rely on self-reports. You can also be creative, try to think of other means that can measure your beat. Okay, so here is an example as to how the author came up uh, with measuring children's fear beliefs. Okay, so how are you going to interpret this if the scores are higher? How are you going to interpret this if the scores are lower? So in this particular set of questions, he devised this tool wherein th- the fear beliefs between the couscous, and coca are determined, okay, by just using some of the components, referring to them, to each of them. Okay. So some caveat, okay, in designing an experiment is that within research, there are a limited number of designs that we can apply. And often there are very different experiments that will conform to certain experimental structures. So again, going back to our discussion about the statement that we can have the same independent variable, but how we manipulate it can vary. However, in terms of number of experimental designs, it's also limited. But in terms of the findings that we can get from it, it is possible that it can also vary. So there are some meta science research going on worldwide where they are looking at how interpretations or the results may vary just because of the experimental design. That's why we need to report everything when we conduct our experiment so that other people can look into it in a broader perspective and try to observe how similar or how different is it from other findings. And what we want to do as a researcher is that we want to collect this evidence, whether each piece of evidence converge to a particular perspective, to a particular direction of thinking. And when we notice that if something is anomalous or something is inconsistent with the theory, we can try to formulate what's wrong. We can try to formulate and move the discussion forward by looking at some gaps that can explain these inconsistencies. Okay, so that's for our chapter 2. Thank you for listening.